My name is Holly Croft. I'm from the School of Biosciences at the University of Sheffield in the UK, and I'll be talking today about the importance of considering plant physiological traits and how we use remote sensing to get at those physiological information in particular, and then how we can use that to sustainably optimize crop yields. So sustainable and resilient food production is key for promoting global food security and reducing climate change. So we all know that the increase in global populations and the more extreme weather events are placing an increased pressure on food production. So remote sensing offers a quantitative assessment of how plant traits vary spatio-temporally, and gives us an opportunity to monitor how plants are behaving in order to sustainably intervene and reduce yield gaps, for example. So when we talk about functional traits, they could be defined as structural, physiological, biochemical characteristics that affect the performance of the plant. And this is a conceptual diagram here by Weiss, it's 2020, that links the kind of physiological structural traits that we can derive from remote sensing data and the type of remote sensing techniques, information that we would use for that specific trait. And then also, if we include meteorological data in that information, then we are able to start predicting crop yields as well. So there's a lot of operational uses of Earth observation data now. So this is an example from GeoGlam, which is an initiative, I think, in partnership with NASA Harvest. And this shows NDVI, which most people have probably heard of. It's the most well-used and famous vegetation index. And it's a structurally sensitive index. So it is a proxy for LAI, basically, or biomass. And on the left, it shows spatially the differences in biomass across the globe. And then on the right, temporal effects, so how LAI might change from year to year, or from one point in time to another. And then if you integrate metadata into this, and we can get a prediction or a monitoring system for how crops are performing at a point in time. This is quite useful for predicting like the impacts of drought or yield loss or famine, or it has economic implications as well for trading structures and pricing strategies. So this is all useful information, but it doesn't. It reflects the conditions when they've already happened almost, or on the way to happening, and it doesn't give us an advanced warning system of how we can intervene from a sustainable management point of view. So this is adapted from a paper by Marlin in 2016, and this really shows the 3 dimensions of remote sensing. So we have to tailor the platforms and the techniques and the sensors that we use to the research questions and to the information that we're trying to derive. So you've got spatial scaling on the X and then temporal on the Y. And what I'm trying to get at here is that no one platform can provide a complete source of information. It's really important to select carefully what kind of data we want to derive. And then the 3rd dimension is the sensor that we use, so the remote sensing information that we are trying to derive. So when we think about plant stress, so that might be nutrient stress, water stress, disease and pests, the first impact on the plant health will be changes in the fluxes. So it'll be photosynthesis or transpiration. And that's the early warning, potential point in time that we have to intervene to reduce the impacts of yield loss further down the line. So if we want to get this kind of like early manifestation, then we would want to think about using thermal or chlorophyll fluorescence data. And at this point, yield impacts will be reversible. We could go along and add more fertilizer, for example. And then the next manifestation of stress will be changes in pigment, so changes in chlorophyll content, for example. And we would use multispectral or hyperspectral data to monitor changes in pigment. And if we intercept them, then there'll be limited damage. And then finally, the structural changes. So when it's propagated through to actually changes in biomass and LAI reduction, we might use structurally based information. So it could be radar, it could be LiDAR, it could be optical bands like NDVI, for example. So the point that I'm really trying to make here is that if we use NDVI and LAI, then we're monitoring biomass, and it might already be too late. So the first, I'm going to present findings from 2 studies today. And the first study is a variable nutrient application piece of research, and this is using drone-based and leaf-level work. So we're kind of quite fine scale at this point. And really the research questions that we started out with was, firstly, how do crop traits differ according to nutrient application? And then secondly, can we detect changes in these traits from remote sensing data? And this image is a little bit of a spoiler alert here. So we can see quite clearly already visible changes in visible wavelengths according to nutrient treatment. So this is a kind of a schematic of the field trial that we used, and really the important information is in the table at the bottom. So we varied nitrogen, sulfur, and sugar applications. So you have a control, so there's 0 fertilizer, and then standard nitrogen, which we also call big N for some reason. It's kind of stuck in some of the later graphs. So that's like a conventional nitrogen application. So all of the nutrient applications, they're of the same amount, but then they vary in terms of how often they're added to the field. So you've got a conventional nitrogen application that's added 3 times, and then little and often, so it's split. So you give smaller amounts more often. We wanted to see if this reduced nitrogen loss and if, increased plant nitrogen uptake efficiency. And then a standard nitrogen with sulfur as well. So we measured leaf level gas exchange, hyperspectral reflectance, and also ACI curves, I should say, carbon dioxide response curves. Ground-based thermal imagery, stomatal impressions, leaf area index, and we took drone flights using an optical sensor and a thermal camera as well. And we're still processing the thermal drone imagery, so I don't have anything to show of that yet, but everything else is ticking along. So this is the kind of biochemical, physiological and structural responses from the different plots to the nutrient treatment. So the 0 fertilizer application is in purple, and then the highest and the most physiologically healthy plots were the conventional nitrogen and sulfur application in yellow. So we can see clear impacts in the leaf chlorophyll content through reduced nitrogen. That propagates through to a lower photosynthesis, and then we can see the manifestations in the leaf area index as well, values. So that's the first point to notice. And the second point to note is that the VCmax and Jmax, so carboxylation and electron transport pathways, remain consistent. So you can see on the regression between Jmax and VCmax. So basically the plant nitrogen allocation strategy held regardless of sulfur or nitrogen application. And we don't have grain analysis yet, but here's just a kind of visual demonstration of the wheat ears. So going from left to right, as left is the zero fertilizer, and then the larger ones in the middle and at the far right are the conventional nitrogen and sulfur. So we can see implications in terms of grain amount, and then we're going to do some quality analysis on it as well. So we wanted to know, can we detect these changes, these biochemical physiological changes using remote sensing? So firstly, we looked at the thermal cameras, the images from the cameras. And then the white that we can see is where the software algorithm has detected the background soil. So all the kind of soil is stripped out, and then it calculates leaf temperature on the remaining leaves. So at the bottom left-hand panel, we've got temperature from the leaves. So we can see that's inversely related to transpiration rates in the middle figure. So basically, the zero nitrogen plants are photosynthesizing less, and there's less evaporative cooling, so the leaf temperature is higher. And then if we look at the regression between stomatal conductance and temperature, we see a fairly a strong relationship there as well. So this indicates that on a relative basis, we're able to detect directly physiological fluxes using thermal remote sensing data. And then we also wanted to have a look at optical wavelengths as well. So this is more geared towards biochemical responses. So on the left-hand side, we've got hyperspectral reflectance. So the zero nitrogen in orange shows a higher green peak at about 550 nanometers, which visually, we saw in the picture early, in the photograph earlier on. And then the right-hand panel is relative reflectance against the conventional nitrogen and sulfur as the best performing physiological kind of condition. So we wanted to see like how we could discriminate the less well performing nutrient applications against that as our kind of healthy baseline. And we wanted to see which wavelength in particular we could use to discriminate that status. So we can see that in particular, we've got the visible wavelengths show the largest difference from healthy status, and also water-based absorption features as well at 1400 and 1900 nanometers. So we use the Meris Terrestrial Chlorophyll Index, MTCI, which uses wavelengths that are that we found to be most discriminating, and we calculated MTCI across the plots, and then compared that to leaf level chlorophyll values that we measured in the lab. We can see a strong correlation. And based on previous work that's shown that chlorophyll content is a strong proxy for VCMax, based on the idea that chlorophyll is a representation of the photosynthetic nitrogen pool. We regressed, we created an empirical model basically to link the MTCI to VCMax, and then we were able to map VCMax across the field site. So I've just highlighted the different nutrient application plots here. So the red is the 0M, and then the nitrogen and sulfur is in the blue. So you can see a clear differential, and then this kind of intermediary status of the little and often, and the conventional nitrogen as well. So this shows like an opportunity to basically map the photosynthetic capacity, which is expressed through the biochemistry changes, and also the fluxes through the thermal. So that's all been done through drone flights, and there's really high spatial resolution. But we wanted to see how feasible deriving this kind of physiological information is from readily available satellite data as well. So if we wanted to kind of operationalize it, if we wanted to scale it to large spatial extents, or in a less kind of resource demanding way, we need to be able to scale to satellites, basically. So this is work from a research campaign in southern Ontario in Canada. We looked at 13 sites across 2 cornfields and 15 sites in 2 winter wheat. Again, it's a variable nitrogen application field trial. So we used the Ontario Ministry of Agriculture and Rural Affairs recommended rates, apart from an exclusion zone, which is indicated on the maps by the white box with the field, and the dots kind of indicate where we sampled within the field. And we measured leaf chlorophyll content, leaf area index, and hyperspectral reflectance. And then we used satellite data. We used Landsat 8 surface reflectance product, so it was already atmospherically corrected, 30-meter resolution, 16-day repeat time, and we managed to get 7 cloud-free dates during our measurement campaign. So when we scale up to kind of canopy level reflectance, which is inevitable from remote sensing data, we have to consider other sources of influence into the pixel, right? So we're now talking about 30-meter resolution pixels. And within the pixel, it's not only leaves, it's also the amount of leaves. So it's leaf area index is integrated, right? And also background reflectance, so how soil and how different solar and view zenith angles also affect that aggregated 30-meter reflectance, canopy reflectance value. So we used a radiative transfer model, which is called SAIL, to model leaf level reflectance from canopy level reflectance, using both dynamically varying variables, so leaf area index varied, and also solar zenith angle, and then we use fixed structural parameters as well, and a lookup table inversion approach to derive leaf level reflectance from the canopy level reflectance. Once we have leaf level reflectance, we can input that into Prospect, which is a leaf optical model, and it's a plate-based model, and that models a number of leaf traits by minimizing merit function, and the most important of those traits to us is leaf chlorophyll content. So this shows the temporal dynamics of measured chlorophyll content, and that's meant to say LAI, across a growing season. And really, I want to kind of point out the separation that exists in leaf area index and leaf chlorophyll. So they have different temporal profiles. So it's really important. If we want to get leaf level information, then we have to remove the effect of the confounding leaf area index values, which are varying independently of leaf chlorophyll content. So using the radiative transfer inversion model scheme that I outlined just 2 slides ago, this shows modeled leaf chlorophyll content from the Landsat data for the cornfields for 4 different days across the growing season. So the white boundaries are the fields, and then the black boundaries are the zero nitrogen plots within them. And we can see there's a nice change temporally as the plants start to go from peak growth to start to senesce. So we're able to detect this physiological information from Landsat data from space. And this is validation against measured leaf chlorophyll content data at the leaf level, which is the top panel, and canopy level, so canopy chlorophyll content at the bottom. So just a quick summary and then some perspectives on how I think we can really make some more leaps in this field. So physiological data is really key for early detection of crop stress or crop health, and this is the only way that we can kind of intervene in the management of crops to close yield gaps. And this requires careful selection of the platform and the sensor in this way. So I think moving forward in the kind of the big leaps that we can make and the direction that we're going in are multi-sensor integration for one, so integrating thermal and hyperspectral data. And this is often constrained by cost and availability of equipment, so particularly when we're talking about drone level, you know, hyperspectral sensors are really expensive, so it's how we kind of mitigate that. I think cloud-based real-time analysis. So again, from a drone perspective, if a drone could fly over a field, as it's taking each image, upload it to the cloud, process it using, you know, advanced machine learning or radiative transfer models, mosaic it, and then by the time the farmer has flown to the end of the field, then it downloads the product, and that, I think, will provide more decision support mechanisms for the farmer as well. Instead of, you know, a basic NDVI map that isn't really helpful, I think we could really advance in these kind of like analysis-based methods, but also like cloud-based integration as well to better, to provide more nuanced remote sensing data. So just to acknowledge mainly my postdoc, so Dr. Bobby Kane, who did a lot of this work and collected a lot of the data. So a lot of this is from him, and also ADAS and CF fertilizers that were responsible for the field trials, and then the support of funders, of course, as well. Thank you.